Hello, my name is Dave Woodle, and my presentation product is on blood. So, the guidelines for this presentation had 8 different topics to be covered, and what I have done is organize these into 3 main areas. What is blood? Some of the key components and characteristics. How does it keep us alive? Looking at each of the components of blood and all the cells in the blood, as well as transport. And then, of course, what happens if we have blood loss? How do we stop it? And so, if we look forward, the vocabulary words that were identified are listed here, and you may want to just take a copy of these and then, kind of note as we go through the presentation some of the definitions and terms. So, what is blood? I think most of us understand what blood is at a high level, but a couple interesting characteristics. First of all, it's identified as a connective tissue, even though in liquid form, which I found is interesting, but the more I think about it, it does make sense as it connects through the entire body. It really monitors things in very tight ranges. The acidity range, the pH range is 7.35 to 7.45. I found that interesting how tight it can do that. We have about 5 liters, 4 to 5 liters in an adult of blood, and we regulate our body temperature at the 98.6 Fahrenheit based on the blood staying about 100. One thing that I hadn't really thought about before until I put together this presentation was that we can't artificially produce blood. We rely on donations of people, that when people need blood, it comes from donated sources. And that donation process does have to have a concern about antigen types. We have an A and B, and so you can have both, one or the other, or none with type O. And then the rhesus factor, the RH factor, rhesus named after the monkey where it was discovered, um, is a plus or minus. So, early on, years ago, hundreds of years ago, that they were concerned about what infections were happening as you move blood around. It really became of incompatibility, and we've learned a lot about that. And I think everyone does understand that only certain types of blood can move back and forth, so there's not an inconsistency in both the RH factor and the A and B antigens. So if we put blood in a centrifuge, it's spinning and it really then moves everything into 3 layers. The lighter plasma is primarily water, and it contains key proteins that are used for a variety of things we'll talk about a little later, and other solutes, which are very important though, like nutrients and ions that move around the body and transport within the plasma. The bottom area is formed elements. Most of it is red blood cells, erythrocytes. But the center area is called a buffy coat, interestingly named because of the color. It's like a buff color. And that contains both platelets and white blood cells. And we'll talk about each of those. But this gives you the percentages of each of the different areas and then percentages of different types of white blood cells or proteins that exist within the plasma. So if those are the characteristics and components, what are the key attributes that help keep us alive? It really falls into 3 categories: transportation, moving things around; regulation, keeping things in line, in balance; and protection. So transportation, as we would know, the main one is oxygen. That is transported everywhere in our body. It's needed to produce ATP through our body to keep energy and keep us alive. But we also have nutrients and hormones that are transmitted. We know how important hormones are to move between organs in the body and the brain, and messengers and neurotransmitters to let us know what functions should occur throughout the body. And we have a lot of reverse path, where carbon dioxide goes back in to be exported out of the body. Regulation, we've already talked about acidity, temperature. Fluid balance is very key, and we have proteins inside the blood to keep the balance of fluid. And then protection, both from infections with white blood cells and clotting, which we'll talk about more. So those are the key functions. If we dig into plasma, as I said, is primarily water in terms of percentage, but in terms of key attributes, the proteins are essential. And these proteins are key in keeping the pressure and level of fluids correct, keeping the amount of water in and out of the blood, keeping the amount of water that's out around the cells, interstitial fluids, keeping transporting antibodies and white blood cells help do that. But we have them moving all through the body and other key proteins and hormones that I mentioned, like let's say insulin. But in addition to the proteins are basic solutes, although very small in percentage, very important, right? Our carbohydrates are really glucose that comes in, helps feed the entire body. We have lipids or fats, and then electrolytes, ions are key for all of our functions in the body, neurotransmission, but also contraction of muscles, for instance. And I've talked about the bidirectional nature of transport, where we have waste products which can be returned for export out of the body. So this gives you some percentages and of where things are. It's a small percent of all these other solutes, but they're key. And as I mentioned, albumin is the major one for controlling the amount of fluid that we have in and out of the bloodstream. So, in addition, then outside of plasma are all the blood cells. We showed that Buffy coat has the white and platelets, and then the red blood cells are a majority, but they all come from bone marrow. And bone marrow is, bones we know give us structure, but they're also key in housing stem cells that produce the variety of blood cells that we have in the body. So hemopoiesis is really the function of taking these stem cells and turning them either into platelets or red blood cells, erythrocytes, or white blood cells. If we focus on white blood cells for a minute, there are really 5 different types of white blood cells. And I found it very interesting that each of those have very unique and distinct functions. It's not just like we have some white blood cells and they go tackle what's happening there. They have specific forms that have very effective functions. We'll talk about this in other sections outside of this presentation, but they can, some like lymphocytes focus on viruses and basophils focus on allergy response, etc. But if we look at red blood cells, which of course are 70% of what makes up all the cells, erythropoiesis is the process of taking those stem cells and moving them through reticulocytes, which are almost a red blood cell, but they only last a few days and they still have some organelles, which they lose and they become the red blood cell. And that red blood cell is key for moving oxygen around, but it's shaped in this biconcave shape that allows it to fold and get through very small spaces that we need at the end of the transport chain and to carry oxygen down to every part of the body, which is done where it's tied to hemoglobin. But anemia is the situation where we don't have enough red blood cells or they aren't functioning properly to carry the oxygen we need through our entire body. So if we move to hemoglobin, that is really an interesting design where we have these 4 folded chains that each one have an iron ion, and that's where the molecule of oxygen is tied to. So we come from the lungs, we move it through the bloodstream, and then it's released to the cells where we need it. At this point, we can actually have carbon dioxide, CO2, connect back and go back to the lungs for export. Some CO2 can be dissolved in the blood too, to be able to move back for the lungs to be able to export then that from the body. So those are the main components that we have and how they function to transport or to protect with white blood cells. One last feature is clotting. Of course, we start losing blood, that has to not occur. So hemostasis is really the process of stopping loss of blood. It starts with platelets. Now, platelets are also formed in this process from stem cells, and they produce a megakaryocyte. Now, megakaryocytes, mega is interesting to me because if you have to look under a microscope, it seems hard to be a mega, but in relative terms, it is much larger. So platelets aren't really a cell. They're fragments of the little extensions that come off of megakaryocytes. They get broken off and they become platelets. And platelets don't have a nucleus. They do have some proteins and mitochondria, but they're extremely critical to the clotting process. What is that process? It's really three steps. If you have injury into a vessel, blood vessel, first of all, it constricts and then it forms a platelet clot. And so those platelets bind together, and then coagulation occurs where fibrin is uh, is created and formed. And I'll show you that in a minute. And it wraps around these platelets and has a very strong clot. But if we look at those steps, we see that platelet activation happens because of tissue factors and collagen that's released whenever you have the injury. As they start to bind, they actually send out for help and they get more recruitment, and all these platelets come together. And then there's a clotting cascade. There are a number of factors, 13 different factors in this cascade of clotting that basically produce fibrin that wraps around as I talked about earlier, the platelets and forms a clot. But then we have to degrade that clot, right? If we kept having a clot, we'd stop all blood flow. So we essentially need to degrade that, and plasmin is key in doing that. So if we walk through this a little further, we see that as platelets are activated because of the clotting factor or tissue factor, it's also called, that they start to go in and bind together. They recruit more of them. And then across the top, we see this clotting cascade that goes through all these factors down to produce fibrin. And fibrin then wraps around, as we talked about, the platelets. This is kicked off with the Wildebrandt factor that's really that clotting factor and allows this whole process to begin. And we form the clot. Well, clotting cascade can be somewhat of a confusing area, but it goes through a number of steps. So either it's based on internal, intrinsic, which is damaging a vessel wall that's maybe caused internally, or trauma where we actually have an injury and we would have an opening into the vessel. And each of those have different factors that go down and activate then this clotting process. So they're numbered by when they were discovered, so they're not in sequence. But factor 10 is produced either from either of these pathways, go to a common pathway, which goes to thrombin, which then activates fibrogen that produces fibrin. So I guess the summary of this is each of these factors are activated by a prior factor, goes through, that's why I call it a cascade. And we can look and see how uh, factor number 12, based on the contact, gets activated. That activates 11, which activates 9, and using calcium activates 8, that produces 10 activation. Or if we have damage, we have tissue damage, we have tissue factor that activates then number 7 factor that also activates 10. So at the bottom line here is fibrin is produced and it stabilizes that clot. How does the vitamin K come into play? Well, that is used back in the liver, where vitamin K is a key factor to create and cause the creation of these coagulation factors. Example here is shown of key factors. So if you don't have active vitamin K, then you won't have the right clotting factors, and you may not be able to do clotting in the body as necessary. And I mentioned already that calcium is used in this process too. So when can there be too much of a good thing? Well, it does make sense. You don't want to have more clotting than you need. So after the clot is formed, plasmin is released. And plasmin is a key capability to start then dissolving this fibrin and start dissolving the clot itself and allowing the vessel walls to be reproduced. And there are cases where we don't want clotting, right? We don't want clotting in certain parts of our arteries around our heart, leading to heart attack and stroke. So anticoagulation is the process of stopping clotting when we don't really want it to occur. Now, that can occur naturally in the body through protein C or S, but it also can occur where we provide medicines. Some medicines prevent platelets, and aspirin is an example. We know that people will take sometimes to keep their blood from clotting. Vitamin K with warfarin is an example to prevent vitamin K from producing, as I mentioned earlier, some of the clotting factors. But this is really the definition of coagulation is where we have clotting. Anti, of course, would be non-clotting. And there are cases where we want to limit that. It's a balance. We need to clot if we have an injury. We don't want to clot, of course, if it's not necessary. So in wrapping up, this is all the key vocabulary words we're to cover. I've put them into where we talked about them in each of the three categories and a list of all the references and wanted to wish you luck in your exam. Thank you.